pleasure to introduce you to Professor Malamaini. Professor Malamaini is an MD, but expert in immunology. She finished the postdoc with Peter Waverley as a T-cell immunologist. She establishes her own lab at UCL in London in 2002, and in 2008 she got her professorship. I had the pleasure to be 3 months with Mala in London this past year, and I think I thought it would be very interesting for you to have a glimpse of what she is doing in her lab. Very good lab, very serious lab, publications in Nature Medicine, Journal Immunology, Journal Experimental Medicine, so I think a very prestigious lab. And the title of her talk, I think it's relevant for this institution because, as you know, hepatitis B, although we have a vaccine, is a disease that affects millions of people of the world. Some areas have a high prevalence, some others not so high, but I think it's a very relevant pathogen. And she's going to explain us from an immunological point of view what can we do to achieve the cure of this chronic infection. Mala, pleasure.
Well, thank you so much for the chance to visit one of my favourite cities. It's absolutely beautiful to come and see that blue sky in Barcelona after the snow in London. And I'm very grateful to have got to know Javier and had him visit our lab. It was a really lucky change in our lives, and I'm very happy that we're going to start collaborating more closely together. And I'll mention some of the potential for that as I give my talk today. Okay, so I don't know how many of you actually work on the liver or hepatitis B, so I'll try and go slowly, and do interrupt me if you have any questions as I go along. It's a very big problem still, hepatitis B, estimated 240 million people infected with this very small, very clever DNA virus, which is hepatotropic, so it just targets the liver, and non-cytopathic, so the liver disease is not caused by the virus itself, but by the immune response to the virus. The result of this liver disease is either hepatocellular carcinoma or liver cirrhosis, still causing an estimated 700,000 deaths a year, so in the top 10 causes of death in the world. And after being neglected for a long time, hepatitis B has started to receive more attention, and for example, this nice publication showing recently in The Lancet led to some headlines even in the tabloid, the mainstream newspapers in the UK at least, saying that viral hepatitis kills as many people as the big killers that everyone pays attention to, like malaria, HIV and TB. And that in fact, importantly, the number of deaths due to viral hepatitis is continuing to increase over the last 2 decades. As is liver disease deaths generally, at least in the UK, the only cause of mortality that's increasing at the moment is due to liver disease deaths. So these are obviously very important problems. And they're very interesting to study because this virus takes advantage of the tolerogenic niche that you have in the liver, which is quite a unique immune environment, important to understand for many reasons, not just for hepatitis B. We know that the chronic persistent infection is perpetuated by a failure of the virus-specific T cells, and therefore there's a lot of attention now being paid in the field to how can we boost T cells to try to gain control of this virus, and also the major complication of hepatocellular carcinoma, which is the second leading cause of cancer deaths worldwide, very poorly treated. And so immunotherapy is considered a very promising way forward, but we need to understand more about how to boost the T cells. So the approach that my lab takes is predominantly based on clinical material and patient samples because, as Javier mentioned, I am a medical doctor. I like the way he said, I'm a doctor but a good immunologist, as if those two are pretty incompatible. Most of our work takes advantage of the access to well-characterized patient samples, and we're very lucky in this disease that we don't only get blood samples, but we can also get biopsies, which are taken still as part of the routine assessment, at least in some clinics. But then more recently, we've also started to get increasing access to bigger pieces of liver from explants, resections, and most recently to the perfusates, the washouts of transplant livers, which turn out to be full of immune cells that are characteristic of the liver. And this is one of the areas where we're now collaborating with Javier and the team, the surgical team here, to obtain some of these perfusate samples. With these, we carry out a lot of immunological analyses, mainly blood flow cytometry, mainly directly ex vivo, so we want to get as close as possible to what's actually happening in the patient, and ideally at the site of the infection in the liver. But we're interested not just in studying single cells in isolation, but rather looking at immune cell interactions, because we know that the immune system requires a lot of cooperation, but there's also a lot of negative regulatory interactions between different cell types, and these are probably exaggerated in the liver because of the anatomy of the liver, where you have this very narrow vasculature permeating through the liver with a very sluggish blood flow. So different immune cells are brought into intimate contact with each other in a way that probably doesn't happen in many other sites in the body. And you also have a very highly specialized both sinusoidal and parenchymal cells. You have these little windows, fenestra, in the sinusoidal endothelium, which means that cells that are just coming through the vasculature can directly contact the infected hepatocytes or stellate cells. And as I said, the immune cells themselves would be brought into prolonged close contact with each other, which then would help to shape immunity. So through these types of experiments, we then are aiming to find molecular targets for immunotherapy that we can bring back to the clinic. And as one step in that, we've just started a little bit of work in the adeno HBV model for HBV, which allows you to do studies in vivo in mice in a model which to some extent recapitulates chronic hepatitis B. And just as an example of the type of immunotherapy that we've been involved in that's starting to happen, we took part in the first-in-man use of genetically engineered T cells for a case of hepatocellular carcinoma as a complication of hepatitis B. And this patient had metastatic disease, but the liver itself was clear of both hepatocellular carcinoma and of hepatitis B infection, so it was a safer case to start trying out T-cell therapy in vivo. Those T cells are engineered to have the right receptor to be able to recognize hepatitis B infected cells or hepatitis B expressing carcinoma. But we're also doing further work to try to genetically modify those T cells to make them, reprogram them so that they can survive better in the liver. And you'll see from the subsequent slides I show you why that's important. So the central problem that we see in chronic hepatitis B is a profound disablement of the T cell response, a loss of T cells, and the few T cells that remain are poorly functional. So you can see that if you look in the liver or indeed the blood of a patient who's managed to resolve hepatitis B, we see a reasonable number of T cells and they're fully functional. And when we start looking in the livers of patients with chronic hepatitis B, there's very few T cells left that are able to recognize hepatitis B. And the ones that are left are not producing many of these effector molecules, the cytotoxic molecules or cytokines that allow them to control the virus. And the key question now is, can we try to recover the T cells and get them back into the state that you see in acute resolving infection? So unlike in something like HIV, where there's really no precedent for immune control, at least we know that in hepatitis B, the majority of adults who get this infection have long-term control of the virus. So the immune system has the inherent capacity to do a very good job with this virus. And even some patients who've had chronic infection for many years do resolve spontaneously. So we know that this is potentially achievable. And the question is, how feasible is it to do this? Is it safe? How are we going to do it, and in whom should we do it? Well, plenty of questions there. And if you want to read more about it, I've just written an article sort of talking about some of the ways that we could consider doing that. The factors that we think are driving the failure of the T-cell response are threefold. One is the high level of infected hepatocytes, and not just a very high level, a very high percentage of them are infected, but remember that it's very long-standing infection. So many patients would have been infected either around birth or as a young child, so they'll have had decades of their immune system being bombarded by this virus. But then on top of the high level of hepatocyte infection, there's also production of very high levels of circulating virus and of subviral particles. So, for example, the surface antigen of hepatitis B is in the serum in microgram quantities. That's probably the only situation in humans where you have that higher concentration of a circulating antigen. And this may contribute to paralysing the immune response. And then you have this special liver environment that I mentioned, because the liver receives its blood supply from the gut, and it's then bombarded with all the gut antigens, food antigens, lipopolysaccharides. It has evolved a tolerogenic environment where immune responses are naturally switched down-regulated, which is good for protecting and preventing overreaction to the gut antigens, but therefore makes it a favourable environment for pathogens to hide out. So what kind of defects do we see in the immune response? I'm mostly going to concentrate on the T cells because they're the ones that are particularly important, we think, for controlling intracellular viruses like hepatitis B, and they're what we mostly work on. But I just want to point out that a coordinated immune response is important. And, for example, B cells and antibody responses are a very important component which need to be studied further. We have shown that the B cells seem to have some pathogenic changes in chronic infection, so there's a skewing towards a regulatory subset which produces the immunosuppressive cytokine IL-10, which can impair T cells, and we have some unpublished data showing an expansion of an atypical subset of B cells, which seems to have some parallels with the exhausted T cell response. And what about NK cells? NK cells are very prevalent in the liver. They make 20 to 30% at least of the intrahepatic lymphocytes, and they have the capacity, in theory, to have antiviral function through either production of cytokines or cytotoxic capacity. Why can't they control this virus? Well, one reason may be that we and others have shown that in hepatitis B they start to have defective interferon gamma production, so they lose the ability to produce this cytokine, which has been shown to be really important for non-cytolytic control of hepatitis B. So because hepatitis B infects so many of the hepatocytes, you don't really want to get rid of it by killing all infected cells, otherwise you could potentially wipe out the liver. So non-cytolytic control through cytokines like interferon gamma, which can clear the virus without damaging the hepatocyte, is a very important mechanism. But the NK cells have a limited ability to do this. They lose their capacity to do this. We recently started to ask ourselves, are these NK cells that are sitting in the liver just a representation of what we see in the blood, or is there a separate subset? And just as a quick diversion, I'll just show you this data that was produced by Kirsten Stegman in my group. And just before I forget, to point out that a lot of certainly all the hepatitis B biopsies that we get are from our clinical colleague Uppy Gill at Barts in London. And what you can see here is that in the blood you have NK cells in this quadrant here, which have got high levels of the transcription factor Tbet and lower levels of Iomes. But in the liver you have these two subsets appearing. The one that you see in the blood is there, but there's also this additional subset which has a different transcriptional profile. So it's Tbet low and Iomes high. And this was seen consistently in all healthy and HBV-infected livers that we examined, and whereas this subset was never found in the periphery. So this is constituting a tissue-resident, liver-resident subset of NK cells, which is uniquely restricted to the liver. And nicely we can mark these cells with a surface antibody, which is important for doing further functional studies, because if you can only look at cells with transcription factors intracellularly, it limits the kind of work you can do. So we found that CXCR6, a chemokine receptor, marks most of this liver-resident subset. And so we can use that to further understand what they do. And one of the things it turns out that these cells are predisposed to is to express the death ligand trail. And previous work that we've done shows that this molecule allows NK cells to kill off virus-specific T cells. So rather than helping to contribute to controlling the virus, these NK cells are actually having a pathogenic effect and deleting the virus-specific T cells that are needed for control. And one of the pathways that this happens through is through trails. So the liver-resident NK cells have this inducible death ligand trail, and the receptor for that is mainly expressed on hepatitis B-specific T cells. It's not found on all T cells, not even all T cells in the liver, but particularly on the hepatitis B-specific T cells. So that provides an antigen-specific mechanism whereby NK cells can delete T cells. What about the T cells themselves? So the T cells are being attacked on all fronts in hepatitis B. They have multiple mechanisms which are limiting their survival and function. So they are highly prone to apoptosis. We showed a number of years ago that's through a molecule called BIM. So they have upregulated BIM, and that might be due to liver priming. And then they're constantly exposed to co-inhibitory receptors like PD-1, CTLA-4, the ligands for which are expressed at very high levels in the liver. And they're also exposed to immunoregulatory cytokines in the liver like IL-10, TGF-beta. But then how can we start to rescue these cells if we know more about the signals like the co-inhibitory signals? Can we rescue them by blocking those? And what Anna Zurich and my group showed was that yes, PD-1 blockade can rescue some responses, but not brilliantly. But if you add in the pro-inflammatory co-stimulatory signal IL-12, you get much better rescue. So here she's combined PD-1 blockade with IL-12, and then you start to get a more consistent rescue in vitro of hepatitis B-specific T cells. And she's gone on to look at the mechanism for that, and it turns out that there's a metabolic defect in the hepatitis B-specific T cells that IL-12 can partly overcome. And this sort of illustrates the importance, I think, of not just looking at surface inhibitory receptors like PD-1, but going to a deeper level in the T cells to try to understand what the fundamental common defects are that we could maybe target in a more direct way. What Anna showed, and I don't know if you've had talks on immunometabolism, but it's a very rapidly growing area. Suddenly the whole field has woken up to the understanding that in order to mount an effective immune response, T cells need to massively increase their uptake of nutrients and also their capacity to utilise those in an efficient way. And so a functional T cell has to express lots of glucose transporters like GLUT1 and be able to take up lots of glucose and utilise both glycolysis and oxidative phosphorylation for maximally efficient energy bioenergetics. And what we did was to compare the T cells in the same patients that were directed against a well-controlled virus for which we use CMV. So many of our patients have got CMV, like a lot of you have, under lifelong control by a very efficient T cell response. So we use that as a sort of ideal model for a good T cell response. And then in the same patients, we looked at the T cells directed against hepatitis B, which represent, as I've shown you, this very exhausted T cell. And in those, there was plenty of uptake of glucose, plenty of the glucose transporter. They could still carry out some glycolysis, but they had very impaired oxidative phosphorylation. So therefore, their energy production was very much limited. And this seemed to be due to their dysfunctional mitochondria. And IL-12 could work partially by rescuing those dysfunctional mitochondria and promoting oxidative phosphorylation. And there's a body of work now coming out showing these mitochondrial defects are underlying a lot of the T cell exhaustion that you see in other settings, in tumours, in other mouse models of chronic viral infection. So this is not unique to hepatitis B. And in fact, another paper came out from Carlo Ferrari's group around the same time showing that mitochondrial dysfunction was a characteristic of hepatitis B specific T cells. They found this by a totally unbiased approach using gene expression profiling, which was very nice that it was confirmatory evidence of what we'd found from a different method. And they showed that if you use mitochondrial antioxidants, drugs like Mitotempo and MitoQ, which have actually been safely used already in hepatitis C patients, they could reverse some of the mitochondrial defects and promote the T cell function. Another way in which metabolism can regulate T cell responses in the liver is through the arginine pathway. This work was done by Laura Pallett in my group. She focused on a cell type called a myeloid-derived suppressor cell, which is capable of regulating T cells through nutrient deprivation. And she found that these cells, even in the blood, are very much expanded in hepatitis B patients compared to control patients. Within these cells, you can see that they're packed full of arginase. So they have very high expression of this enzyme, which is able to catabolize arginine. And you can see this by flow cytometry here, but also using the image stream, you can see the cytoplasmic expression of arginase 1. And then even in the patient's serum, you see an increase in circulating levels of arginase and a depletion of the amino acid arginine, just even detectable in the serum. But obviously we would anticipate that would be more dramatic in the liver at the site of the infection. And in fact, if we look at the accumulation of myeloid-derived suppressor cells at the site of disease, using these, again, paired blood and liver biopsies from hepatitis B patients, you can see that almost every case the frequency is increased in the liver, the expression of the immunosuppressive enzyme arginase is increased, and their degranulation propensity, as marked by CD63, is also increased. So they seem to be more active and accumulate locally in the liver. So, do you have this game in Spain, or is this an English thing? Tug of war. So I see this like a tug of war over arginine, where there's a strong competition. And I don't like to be sexist, but I think I can see which side is going to win here. So the T cells tend to lose out. In fact, there's not just the MDSCs that can produce arginase and break down arginine, but also hepatocytes themselves, of course, are full of arginase. And if they are damaged and inflamed, they can also break down arginine. And then hepatocellular carcinoma is actually auxotrophic for arginine, so it's dependent on taking up arginine from its environment. So all these different liver-specific cells will be catabolizing and consuming arginine. But why should T cells care so much about this particular amino acid? I mean, there's plenty of other amino acids. What's so special about arginine? And that was only really understood very recently from a lovely work done from Roger Geiger in Antonio Lanzavecchia's lab. And what he found was that T cells actually express another enzyme called arginase 2, which can also catabolize arginine. And so as soon as they take up arginine, particularly if they're very active, because of all their intracellular arginase 2, it gets broken down. And they showed that arginine is required, again, for oxidative phosphorylation and effective metabolism, and also for good mitochondrial function. So there's a very dynamic situation, and in fact, arginase and tryptophan are the only enzymes known to be regulated within the body, the only amino acid enzymes, I should say. And so that suggests that those two pathways are probably the most important ones in terms of metabolic control of immunity. So what effect does this have on the T cells? Well, we noticed back in many years ago in an original paper that we did that if you took arginine away from hepatitis B specific T cells, they really suffered. So here you're looking at the CFSE division of T cells upon stimulation. So this is marking them with a tetramer, HLA peptide tetramer, allows you to identify the HBV specific T cells. And you can see that as soon as you put them into arginine-free media, you have very few responding cells left. And that's because they've not been able to divide, so their division becomes arrested. And you can see there's a sort of dose response here according to the amount of arginine in the media. So this was just an in vitro demonstration that they were highly dependent on having sufficient arginine. And this is sort of concentration where we were starting to see it, this is where our patients lie on this steep bit of the slope. And we also noticed that the T cells directly taken out of our patients had reduced levels of the CD3 zeta chain, which is one component of the CD3 signaling complex. And this is thought to be a hallmark of arginine deprivation. So there was some suggestion that this was going on, but it was only when we did these experiments that Laura did with the myeloid-derived suppressor cells that we were able to show that they were a cell type that was responsible for this arginine deprivation, because when she added them into co-cultures with CD8 T cells, this is looking at the response to a pool of peptides for CMV, EBV and flu, but you can do the same with hepatitis B T cells. As soon as you add the MDSCs in, you get suppression of those responses, both CD8 and CD4. But if you pre-treat the MDSCs with a drug called NOR-NOHA, which specifically inhibits arginase 1, then you can start to recover the responses again. So at least part of their capacity to suppress is through this enzyme pathway. So what I've shown you is that in the liver we think there's multiple reasons why T cells will be starved of arginine, and this then impairs their capacity to proliferate, survive and mediate antiviral function, and probably also drives mitochondrial defects. Okay, so to come on to some more sort of clinical considerations. Will it be safe to try and boost T cells in hepatitis B? This is a big topic of conversation with lots of pharmaceutical companies at the moment. The problem is that there will always probably be a trade-off in this disease between immunity and immunopathology. It's very difficult, and any clinician who's looking after hepatitis B patients knows that, you know, when you see a good immune control, that's usually accompanied by a flare of liver disease. That's, again, as I said before, because the virus itself is non-cytopathic, but the immune response is what's causing the liver disease. So if you need a strong immune response to control the virus, you will probably have to have that at the expense of some damage to the liver. So perhaps hepatic flares are an inevitable result of immune boosting. What can we do to try to control that? Well, minimizing antigen load seems like a good idea. So understanding more about the extent of infected hepatocytes would be very useful, so we could have some better understanding of what reserve we would have in the liver, even if a lot of infected hepatocytes were killed. And obviously selecting patients with good liver reserve for immunotherapies, trying to focus any T-cell boosting on the component that's directed towards HBV makes sense to obviously try and avoid autoimmunity, and then developing adjunctive approaches that we can use to try and shut down collateral damage would be very helpful. Which patients should we be targeting? There's a lot of debate about this at the moment, and there's no clear answers to this. And it may vary depending on which therapeutic strategy we're considering. So some strategies might be better suited to one group of patients or another. Most people would suggest at the moment that it would be a good idea for patients to be suppressed on antivirals or have had at least e-antigen seroconversion before trying any immunotherapies. Another group that's of interest is low-level carriers who are already almost there. They've done 90% of the job, maybe very low S antigen, maybe just some little kick could really clear their infection. But on the other hand, those patients have very low risk of getting liver disease and complications. They're totally asymptomatic, so would they accept potentially risky treatment? There's another school of thought arguing for considering treatment earlier on in the course of the disease. So the first phase of infection, where patients are known as immunotolerant, well, no, we're not supposed to call them that anymore, but they're in the first couple of decades of life after infection. They typically have very, very high levels of viremia, but no liver disease detectable. There's some data to suggest that these patients actually are already getting integration of the virus, therefore putting them at risk of carcinogenesis. So it's not a totally safe phase of the disease and should perhaps be considered more seriously. Of course, they're a major pool of infection because these are often young sexually active patients with very high viral loads. There is some evidence that they may start to even get subtle liver damage, and the immune system should be less exhausted because they've had the disease and the infection for less time than a patient in later stages of the infection. So there are arguments for thinking about trying to treat these patients. And then there's patients who've already got complications, like hepatocellular carcinoma in particular, who are very good candidates for new immunotherapy trials because all else has failed. There's nothing to lose by trying something potentially risky, and then if it looks relatively safe in those, you can extrapolate back to earlier disease. And this is, for example, what's been done with nivolumab, with PD-1 blockade. Now, what about the need for liver sampling? So this is what I just want to end the talk with, is explaining why I think it's so important that we continue to sample the liver while we develop therapies leading towards functional cure. Because in the field, diagnostic biopsies, which have been used routinely as part of the management of these patients, are starting to be replaced by non-invasive tests of fibrosis, etc. But the problem is that the virus itself is not all detectable in the circulation. So the key component of the virus, which is really critical for maintaining chronicity, which is this cccDNA, it's an episomal form of the virus which sits within the nucleus of the hepatocytes, needs liver tissue to be detected and quantified. I mentioned also that the virus can integrate into the genome, and that's what can drive carcinogenesis, and that can only be detected by sampling the liver. I've shown you that we have a subset of resident NK cells which are not in the blood at all, and have particular properties, for example, the capacity to mount a rapid defense against pathogens by trail expression. And then I want to show you now that not only are most of the hepatitis B specific T cells compartmentalized in the liver, but they're actually resident in the liver, which means again that they stay there long term and they can't leave and recirculate into the blood. So we're missing a lot of the action by just sampling the blood. And these resident populations, there's an increasing amount of exciting work coming out from mouse models and other human studies showing that they're really critical players in frontline immunosurveillance. They're the cells that are there ready to mount the first response and damp down any sign of reactivation. So if we're trying to achieve a functional cure where we want to keep traces of virus, assuming that the new antivirals are not going to be able to eliminate every last trace of virus, if we need an immune response there as a backup to keep that remaining trace of virus under control, we really need it at the site where that's going to happen. This is work again by Laura, who does a fantastic job in the lab. And she was able to show that there is this liver-resident population of T cells in human liver. So tissue-resident T cells are typically identified by the expression of these two markers, CD69 and CD103. And you can see that in the blood there's really no cells that co-express these molecules. But in the liver there's a really nice population. This is just now in healthy liver. And we have 3 different sources of healthy liver. We have some biopsies of transplant livers, margins around colorectal metastases, and the perfusates, the washouts from the livers. And in all these cases, you can see roughly, I mean, there's a bit of quite a range, but there's around the same 5-10% of the total T cells that have this resident phenotype. And this is quite heroic work because there's 54 fresh liver samples. Typically, these will arrive towards the end of the day or late evening and then require several hours of work. So this is really a lot of effort. So not only do they have the unique phenotypic expression, but they also have a particular transcriptional profile, which you don't see in the other subsets like the circulating memory T cells. I should have said, sorry, these are all memory T cells, so they all have down-regulated CD27. They have low levels of T-beta-neomas but high levels of BLIMP. And so this is showing a multi-dimensional analysis using TISNI to identify the population here of liver-resident T cells and with their transcriptional profile. So that the 2 markers that I mentioned that we use to identify them play an important role in tethering those cells in the liver, we think. CD69 has been shown to be a negative regulator of S1PR1 mediated T cell egress, So although it's a well-known activation marker on lymphocytes, it actually also plays a functional role when it's in a tissue, where it allows this system of egress to be counteracted, so that it arrests the cells there. And then CD103 is the alpha E beta 7 chain of integrin, which binds to this molecule E-cadherin, which can be expressed by a lot of epithelial cells. And you can see here that the hepatocytes express high levels of E-cadherin. So this is co-staining with cytokeratin and E-cadherin, and then plenty of this red staining, which is the E-cadherin, which you can see here, which may be able to help to tether the tissue-resident T cells within the liver. They also express other homing markers that we know are important to the liver, and in addition they have some functional adaptations that we're currently exploring that may allow them to survive and respond differently within the liver environment. So they have high levels of CD98, which is a good marker for the transporter for some particular group of amino acids, which we know are very important for T cell function. And they also have, interestingly, high levels of the TLR4 co-receptor CE14, which we're currently exploring. But then they have, as you might expect, high levels of, again, CXCR6, like the NK cells, which is this liver-homing chemokine, and CXCR3, which is another well-known liver-homing chemokine. Then so this was all in healthy liver. Now moving on to hepatitis B liver, we again see, and here in this case we have the paired samples with blood and liver, so it's very nice because you can see very clearly that there's no tissue-resident T cells coming out into the blood, even when you have a disease in the liver and inflammation, they're still stuck there in the liver, and there's very variable but consistent expression of these cells in the liver. And overall there's a striking increase 2 to 3-fold in the hepatitis B infected livers compared to the control healthy livers. But this increase is really only seen in the patients who've got good control of hepatitis B. So if you look at here, you can see that in those who've got less than 2,000 international units of hepatitis B, there's a significant increase compared to healthies. But once you start to get to high viral loads, there's no real increase. So that's suggested that the tissue-resident T cells are able to play a role in controlling hepatitis B, which would fit with all the literature that's around from mouse models. And consistent with that, we do see that there's an enrichment of hepatitis B-specific T cells within the liver-resident fraction. So this is all just done on global tissue-resident T cells, but here using HLA peptide dextramers again, which identify the HBV-specific T cells, you can see that quite a large proportion have got this double positive phenotype, and then majority of the remainder are CD69 single positive, which we think are also mainly resident. It's not a totally fail-safe marker for residency, but it will mostly contain resident T cells. Now, these T cells that are living in the liver long-term have very high levels of PD-1, which we know is a molecule able to constrain T cells and normally limit their functionality. So PD-1 is heavily expressed on all T cells in the liver, but it's particularly heavily expressed on the tissue-resident subset here in black, both in the control liver and even more so in the HBV liver. And then if you look at the MFI of PD-1 expression, that really, again, goes up very dramatically in the HBV-infected tissue-resident T cells. And the ligand is known to be expressed by many cells in the liver. Here you can see some staining showing expression of the ligand by the Kupffer cells. So we would expect that then to be really constraining the T cells. But on the other hand, we're saying that they're able to control the virus. So how is that paradox resolved? And this sort of fits with a lot of emerging data recently, suggesting that PD-1 has this dual role. It's not just a bad thing that's limiting and constraining T cells. It's also playing a protective role. And this sort of first emerged from some work from John Werry's lab, using the LSIMV model, where he showed that genetic knockdown of PD-1 actually promoted terminal senescence over differentiation of the T cells. So in fact, PD-1 was important to protect those T cells from undergoing too many divisions and becoming senescent. And we have similar data from Itzio Otana's work in my group using human T cells. So this is all leading to this concept that PD-1 actually is important to preserve T cell responses in the setting of a persistent viral infection where you've got so much antigenic stimulation, that in order to adapt to that and allow the T cells to survive, they utilise this mechanism for limiting their proliferation and maintaining proliferative reserve. But in that case, are they actually able to maintain functionality? Because there's no point preserving them if they can't then do anything because they're so tightly constrained by PD-1. And this was the surprise, was that when we stimulated these T cells directly ex vivo for just 4 hours, in fact they were highly functional, much more functional than the non-resident T cells. Or certainly than, well, in terms of interferon gamma, you can see they've got about the same level of functionality as the non-resident fraction, more than the blood. But when we started looking at their IL-2 production, that's when we saw really striking differences, because IL-2 is normally very difficult to detect from T cells in the periphery. We virtually see none produced. And same with the liver-infiltrating T cells. But the liver-resident fraction have really dramatic production of IL-2. And this is all, just remember, just within 4 hours of stimulation. So instant capacity to produce IL-2. And Anna Zurich in our group had previously shown, when she worked with Percy Knoller, that IL-2 is able to actually overcome PD-1 mediated hepatic tolerance. And IL-2 production by CD8 T cells has been shown by a number of pieces of work to be really important in maintaining T cell functionality. And then here I just wanted to show you this, I've got time, yes. This result, because this is quite unusual, that we were able to get enough cells from a patient who'd resolved hepatitis B. So natural resolution, like I showed you at the beginning, is the gold standard of what we're really trying to mimic with all our immunotherapies. It's the patients who've managed to keep this virus under long-term control. We know that they don't eliminate hepatitis B. There's traces of it left, because if these people get heavy immunosuppression, they will reactivate. But outside of immunosuppression, they will maintain lifelong control of the virus. So this is what we're actually trying to mimic. But it's been very difficult to study in the liver, because you wouldn't normally be able to sample people who've resolved an infection. But some of these perfusates of transplants will come from people who've naturally resolved hepatitis B. And the team here have managed to identify 2 of these for us. And we had one from the Royal Free here, where we were able to get enough cells to stimulate with all the different pools of peptides spanning the major HBV proteins. And you can see this really robust T cell response. So more than 10%, more than 20% actually of the intrahepatic T cells are able to respond to these different HBV peptides in that ideal setting where you've got long-term control of the virus. We don't know how long ago the patient had resolved the infection, but to some degree they obviously have persistent, long-lived T cells able to mount a very efficient recall response within 4 hours of restimulation. And again, if you look at the IL-2 production, most of it is within this resident subset, CD103, CD69 double positive T cells. The next question we asked was, is it possible to artificially derive this liver resident phenotype, which could be useful for immunotherapeutic studies? So if we take circulating T cells from the blood which don't have this double positive phenotype, can we induce this with a particular cytokine profile? And there was a little bit known already about cytokines that are good at inducing tissue residency, and so we had a sort of list of candidate cytokines. But what Laura and Jess found was that it was a sequential signal that was really optimal for inducing this, not one particular cytokine on its own. I mean, IL-15 on its own was able to induce the chemokine homing receptors, CXCR6 and CXCR3, but it was only when T cells were stimulated first with IL-15 and then with TGF-beta, and not with the cytokines in the other way round, that you started to get this convincing population of double positive tissue resident T cells. So this sequential IL-15 TGF-beta signal, and these are both cytokines that are prototypic liver cytokines that they may well be able to encounter in a liver environment, should be able to induce this resident phenotype on circulating T cells. So just to summarise this work, we've defined a signature of tissue-resident CD8 T cells in human liver that can't be sampled in the periphery. They expand in hepatitis B, particularly in patients with good viral control, suggesting that they may be important for a functional cure. And we've defined some of the features that instruct their retention, survival, rapid and non-cytolytic antiviral function, and signals that can recapitulate their induction. So we would suggest that future immunotherapeutic attempts to functional cure in hepatitis B should be aiming to induce these long-lived tissue-resident memory T cells to provide a frontline surveillance. And we would also suggest that liver sampling is really critical in studies of functional cure to detect these and other important local immunological and virological parameters. If you want to read more about this topic, there's the paper and the review. And this is a review we've just written about the importance of sampling the liver and starting to discuss the possibility of using fine needle aspiration as opposed to liver biopsy as a way of longitudinally sampling the liver compartment. And I think that's it. So I'd just like to thank my fantastic group who are really amazing and hardworking. And I've mentioned the individuals as I went along. All our collaborators, particularly our clinical collaborators, and the funding bodies, and I'm happy to take any questions. Thank you. I took out those slides, but we did some longitudinal studies. Well, first of all, we looked at the chronic infection broken down according to stage, and what we found was that the expansion of myeloid-derived suppressor cells and the associated drop in serumarginine was only seen in patients who don't have liver disease. So the predominant evidence was actually that it was sort of down-regulating liver immunopathology rather than an association with viral load. But when we looked in longitudinal samples from acute patients, we could see a very nice inverse correlation with viremia. So as viral load started to come down, the MDSCs also came down and the arginine recovered.
That was in acute patients?
Yeah. There's a little bit of work with chronic flares as well, a couple of patients. But it's limited. I mean, it would be, of course, fantastic if we could look in seroconverting patients. And we haven't looked, for example, in treatment-induced seroconversion. That would be nice. That's a really interesting scenario, which I've been talking about with Javi. I mean, I really don't understand what's going on there. I think the most obvious sort of idea would be that it would be innate regulation that's changing, that HCV's driving innate cytokines that have antiviral capacity against HBV, and when you switch that off, HBV comes back up. But I find it hard to understand how there could be an interaction at the level of the CD8 T cells, but maybe there is something more to it. We've just started to look in HCC resections. In terms of you asking about the tissue-resident T cells, they do get into the cancers in variable proportions. Whether they have the same functional capacity, we haven't yet had a chance to look. But there's a recent paper I think it's from lung cancer, showing a very nice correlation between the frequency of tissue-resident T cells in the lung and the outcome. So that does suggest that they're protective in human tumours as well. Yeah, I mean, unfortunately, most HCCs are not biopsied, so our studies at the moment are limited to resections and explants, but we would very much like to do more of that. Yeah, it's not known. I mean, generally, the whole issue about where HBV-specific T cells are primed is, again, a lot of debate. There's certainly work showing that cells within the liver can prime a T cell response. Both the hepatocytes themselves and the liver sinusoidal endothelial cells have been shown to be able to prime T cells, but then to drive them towards a tolerogenic and pro-apoptotic phenotype. So Patrick Bertolino showed, in fact, that hepatocytes, this is not in the context of hepatitis B, but just in a model antigen, if the T cells are primed by the hepatocytes, they have this BIM high pro-apoptotic phenotype, which is exactly what we see in the hepatitis B specific T cells. It's very difficult in human studies, of course, to be sure where the priming is occurring. Others would argue it's local lymph nodes. It's hotly debated. I mean, there is some work suggesting that Tregs can play a role in liver regulation, certainly in hepatocellular carcinoma, definitely. But in a healthy liver, we've started to look, there's not that many of them there, far less, for example, than the MDSCs. But you're right, that when we saw this IL-2 production, we did start to wonder whether maybe just locally around those tissue-resident T cells, you have an accumulation of Tregs. And we'd like to look at that with some immunostaining and see whether, at least in the vicinity of a tissue-resident T cell, do you have an accumulation of Tregs? But we don't have that information yet. Yeah, thank you for that question, because actually I'm just thinking last week that we need to look at this again, because the paper's just come out, I can't remember which journal, showing a particularly high frequency of myeloid-derived suppressor cells in the neonatal period, in another setting, not in hepatitis B. So that's very plausible, and it would be very interesting to try and get samples to have a look at that. Yeah. It is a very important point to pursue, because to our surprise, it seems like the tissue-resident T cells in the liver are mostly living in the vasculature, in the sinusoids actually. They're not going into the parenchyma. I mean, we haven't done this thoroughly. We've done a little bit of immunostaining, but we see, for example, that we get, if we do these perfusate washouts, which shouldn't be getting cells from the parenchyma, it's not exact, but there's plenty of those tissue-resident T cells in the perfusates. So, and in the mouse data, in the liver, they found that the vascular spaces is where the tissue-resident T cells are hanging out. So the LSECs are the primary candidates, apart from the Kupfers, that should be influencing them. Although, of course, that through the fenestra, they could also be influenced by hepatocytes. I mean, that is being explored also in HIV because of the work in LCMV. So it's not probably unique to hepatitis B or the liver. Probably recently activated T cells are susceptible to this mechanism. And we're trying to look at it at the moment in the HBV adeno mouse model, where it does seem that depleting NK cells can have this effect. The question clinically is whether there could be a safe pathway that you could interrupt without having other downsides of losing NK function. But I do think, I mean, it's the same as, for example, interrupting PD-1. I mean, it will have some potential risks of immune activation, but it could potentially be done in a short-term setting, accompanying a therapeutic vaccine, for example. True. So, but I mean, what was interesting was that the mean number of these cells was the same in all those different types of samples. I mean, there is a lot of variability, so it could be that the degree of ischemia is influencing it within those cohorts. We haven't really been able to look at that. But they're not totally healthy, I realise that. So.
Okay, if there are no more questions in Madrid, thank you, Madrid. And here I think there are no more questions. So again, a pleasure to have you here. I think it was very helpful for everybody. And see you next seminar. Thank you very much.